Hi, everybody. Good morning. Welcome to Lunch with a Scientist. I'm Leah, and I am here with Nicholas King. Hi, Nick.
Hi, Leah. It's great to be here with you today.
Thank you so much. Nick, where are we talking to you from?
I am joining you from Tucson, Arizona today. I work at the Critical Path Institute, and as most of us are, a lot of us are right now, I'm still working from home and have shifted around in the space, and so now my bedroom is my office, and we've made it a little more permanent in that I've brought home my actual desk chair now. So I think I'll be working here for a while.
That's great. Have a good working space, and the desk chair makes all the difference.
Mm-hmm.
So, as Nick said, he's the scientific program manager for the Biomarkers program at the Critical Path Institute, and we are going to hear from him in just a little bit. Before we turn things over to him, we want to thank the Soroptimist International of Truckee-Donner for sponsoring our online learning this week. They have a mission to provide women and girls with access to education and training, which they need to achieve economic empowerment. And so thank you so much, Soroptimist. We are so grateful. This is their 100th anniversary of being in existence. They were founded in Oakland, California. And the local chapter that gave to us this year is celebrating their 50th anniversary. So we're so thankful for this organization and everything they do for the community. And if you are listening and you have a business that wants to sponsor one of these Lunch with a Scientist talks, just let us know, and we'd be happy to send you our sponsorship form. So now to turn things over to Nick. He graduated from the University of Arizona College of Medicine's Cell Biology program with a master's degree in 2009. And today he's going to talk about working collaboratively with scientists and healthcare authorities in the medical field to advance medical innovations. And he works in regulatory science. And if you don't know what that is, you probably will by the end of his talk today. So Nick, I'm going to turn it over to you, and we'll let you get your presentation up and running.
Great. Thank you very much, Leah. All right. Well, I've titled today's presentation Consensus-Based Collaborative Research and Working with Health Authorities. It's a bit of a mouthful, and I'll kind of split this up piece by piece as we go through. It's my pleasure to be here with you today, and a little bit of what I'll be going through. I plan to touch a little bit on my career path. I have a way that I like to go through it. It's a little bit, we'll see what you think of it. And then as well, talk about where I'm employed, the Critical Path Institute, and the work that we do that is consensus-based collaborative research. And finally, research leads into working with health authorities in different paths of drug development and in a non-competitive way. So first, just a little bit about me and where I've been over time and how I came to be at my current place. I was born in Fort Campbell, Kentucky, and soon after that moved back to where I grew up most of the time. And I like to kind of throw in the books that I was reading throughout that period. So you'll see a mixture of pieces as I was growing up. Lafcadio, some good Shel Silverstein, Don Quixote was one of my favorites, as well as I fell in love with the author Graham Greene. Our Man in Havana was a big one. I won't go through all the books, but these are three of my favorites right here. And then as well, I like to kind of throw in the sports I was doing at the time, because I'm not just a scientist. I have other interests, and we'll go through those pieces. I spent a few years in Boston rowing, continued my similar author passion. And then I spent some time in Gainesville, Georgia after the 96 Olympics, coaching at what had been the racing venue there. Moved then to Pullman, Washington, where I got my undergraduate degree in biochemistry and with the emphasis in molecular biophysics. A little bit after that, my wife and I took a trip into the Peace Corps. And while I taught there in English as a second language to high school and middle school students, as well as chemistry teachers. I had quite a bit of free time, so there were quite a few more books I read during that period. And following that, we moved back to Pullman for a brief period of time. And I started graduate school there in Pullman at Washington State University. and then completed my studies in Arizona, at the University of Arizona, where I conducted research in cell biology, primarily looking at how muscle, and particularly cardiac muscle, works to contract and relax, and more on the relax part because it's not an active piece that's driven by energy, but more of a spring component. And following graduation, I'd spent several years working at the bench, and a lot of that I spent working at a microscope in what were frequently rooms where I was with myself and a microscope and fluorescence for 8 to 10 hours at a time. And coming out of that, I decided that I appreciate and enjoy working at the bench, but I wanted to explore how I could continue my work as a scientist, but not necessarily work in a lab. And so I found this institute here in Tucson called the Critical Path Institute. And so we, here at the Critical Path Institute, I started out as a project manager and then worked my way through a few different titles and positions over the past decade. And at Critical Path, what are we? We are an institute that works as a catalyst. So we try and lower the bar for advancing different types of science. And in this case, we're working on developing new approaches to advance medical innovation and regulatory science. And we go about that by bringing together teams to share data that they've collected, knowledge and expertise that we then vet and analyze to come up with sound consensus-based science. And so the consensus component of this is that we bring together, in the case of most of the work I've been, I'll get a little more in detail farther on, but it's bringing together companies that have a common issue that no one company can solve in their work. And so working together across companies in what we call a pre-competitive space is the manner that will advance the science for everyone involved and permit new and exciting developments, and in my case, in drug development and assessing how to monitor safety of drugs. And so through CPATH, we have two of the primary pieces that we work on are regulatory interactions with the agencies for what is called a qualification and either biomarkers or clinical outcome assessment. We have other work that we do. I'm not going to focus on that piece today. And so, we, again, kind of going through a little bit of what we are, our vision and our mission and our values, all these very exciting components to hear about, but lays the groundwork for what we're doing still. Overall, we are working to accelerate the path to a healthier world with through this research that spans companies, it spans patient groups, and also involves working with regulatory agencies. And we are an independent and trusted partner, so we've always maintained our neutrality in this. We are not a drug developer. We are not a regulatory agency. We work in between to bring those different entities together to work on common problems. And so we accomplish this through working on acting, as I said, as a neutral third party, and then as well, we bring together consortia, and so those consortia are made up of industry. For the most part, that industry in this institute are pharmaceutical companies and bioanalytical companies. We work with academics, so different professors and clinicians, as well as different government agencies. As I mentioned, regulatory agencies in the U.S., that would be the FDA. I'm sure everyone here is hearing quite a bit from the FDA right now. We also have partner with the NIH, and part of that is also working with patient groups so that we ensure we have a well-rounded perspective from all the stakeholders involved. And so what we do is we act as that pre-competitive neutral ground where companies can come together. They share data that each component of what they share contributes to a larger project, and I'll give some examples a little bit later on of how I do of what those are. And then we take that data set as a whole and conclusions we've drawn from it, and submit those to the regulatory agencies, so the FDA, for acceptance or endorsement from them in order to use those in future drug development programs that would then permit either a better way that's more specific or sensitive to detect drug injury, or also to demonstrate that while that there is not a drug injury present due to the specificity of what we're measuring. Other components that we work with are something called a clinical outcome assessment, and that is when there are different types of clinical outcome assessments, and on the next slide we'll kind of touch on all of those. So clinical outcome assessments, that is it's a measure that describes how a person feels, functions, or survives. And there are a few types of those that are out there. There's a patient-reported outcome, an observer-reported outcome, a clinician-reported outcome, and performance outcome measures. And so a patient-reported outcome would be a set of questions where, might be something, for say, mental health, it may be asking how you feel that day, or what time did you get up and what time did you go to sleep, how many hours did you sleep, and different pieces like that. And then an observer would be not the patient, but say a partner or a parent or a sibling who has observed that person and can then provide information. A clinician, your doctor, would be providing the report. And then performance outcomes, that's when you're measuring actual movement from someone. So that might be looking at how well you can walk in a straight line if you have, say, some type of muscle disease or degenerative muscle disease. What I'm going to be focusing a little bit more on today are biomarkers. And what biomarkers are is this is a nice long explanation here, but what it really comes down to is measuring normal biological processes, pathogenic processes. So that's when something is injurious or hurts part of your body or biological process, or a response to an exposure or an intervention that might include a drug treatment. And so, some of the ones that everyone I think has probably seen before, you may find relevant, is when you go to the doctor and they take your blood pressure. That's a biomarker to show how well your heart is working, as well as the heart rate. And then as well, when they draw blood from you and they send that off, and then they come back and they may say, well, your liver is looking healthy based on this information that we have. And then another, biomarker would also be an eye test, right? When you can read off a chart and see how far down in the little letters and numbers you can discern. And so with that definition of biomarkers in mind, I have spent the past 10 years working with a consortium called the Predictive Safety Testing Consortium. And what we do is we work on translational safety strategies. And I'll explain that a little bit more further on. We were started in 2006. And what we do is we bring together pharmaceutical companies to share and validate innovative safety testing methods. And once we test those methods and are confident in how they perform to detect a specific type of injury, say that is a drug-induced injury to the kidney or the liver or to your skeletal muscle or to your heart, we then submit documents that summarizes our conclusions and provides the supporting data, and submit that to the FDA, the EMA, and the PMDA. And we are currently made up of 14 corporate members, and all of our goal is to improve safety testing approaches and methods. And our primary route, as I mentioned, is that we take this summary and conclusions from the data sets we gather and submit those to the FDA for regulatory acceptance by them for use. This just briefly shows you who are the partners that we work with, and so we have many consortium members. We consider partners as well the EMA, which is the European equivalent of the FDA, and then the PMDA, which is the Japanese equivalent of the FDA. We also work on several projects. One is with a group in Europe that's part of the Innovative Medicines Initiative. We have also worked with a group called the FNIH, which is the Foundation for the National Institute of Health. And as well, we work with the Japanese equivalent to the NIHS. And so with that piece, kind of, when I mentioned translational safety, what I kind of mean by that is that when you first start working in drug development, you frequently come up with a compound, and then it's advanced for testing and looking at how it is safe or how it is effective in different animal species. And when we conduct these studies, we also have the capability to take information from their organs and assess the actual injury to each one of those organs. In humans, we aren't usually able to take those types of biopsies or slices from the different organs. And so what we're looking to do is to find first biomarkers that measure different types of injury to the liver or the kidney, and match that to the injury that we see in the organs of animals, and have that same biomarker be produced in humans, and look at how, if a human is injured, how that biomarker changes in humans when that specific organ is injured. And we focus on several different organ or tissue areas. The kidney is an important one. There, when we first formed, this was our first focus point, because the ways that we currently, and is still the primary accepted manner to monitor and detect kidney injury, you don't see changes in those proteins or biomarkers until you have a loss of function to the kidney. So your kidney isn't working correctly. You've lost half of your kidney function. So we have two kidneys, and you basically are working with one kidney at that point. And so what we said is, we had a group of scientists across several companies that had very similar sets of data that they had collected, and wanted a place to bring it all together and submit to the regulatory agencies and say, these new types of biomarkers are first more specific to the kidney, and they are also more sensitive. And by sensitive, meaning that before there is significant or major injury to the kidney, we see a change in that biomarker. We also focus on looking at skeletal muscle injury, liver injury, the vascular injury, so your veins and your your veins, and then also the pancreas and testicular toxicity. So transitioning from there with the translational science and safety science that we're working on, I wanted to briefly touch on the major health authorities we work with currently. It's one of the fun pieces of what I do, is that I have a job where I interact with a lot of different people, and a lot of what I do are actually calls similar to what I'm doing today with you. We will have a group of scientists on a call, and we'll discuss topics. As well, I have the opportunity to travel to several of these locations when we have formal meetings with these agencies. So first, there's the U.S. Food and Drug Administration, the U.S. FDA, that is our health authority that we utilize here. In Europe, it's the European Medicines Agency, and they're located in Amsterdam. And the third one is the Pharmaceutical and Medical Devices Agency for Japan, and they are located in Tokyo. These authorities, what their primary purpose is to review, approve, and regulate drug and device use by the public. And so we work with them not on the drug and device component, but on ways that you can monitor and detect how drugs are impacting a person. And so we do this by looking at these biomarkers, what are, as I kind of mentioned before, a simple one to think of is your blood pressure and monitoring that if you have high blood pressure or low blood pressure or regular blood pressure. So when we want to advance something, we have a set of criteria that we put forward before we move forward with a project. And so the three primary that we look at is what is the biological plausibility for this to be used in a specific manner. And so by biological plausibility, is it relevant to the organ that of interest that may be injured? And is it specifically produced or expressed in that organ? And where else might we find it? And then next, once we have established that it is biologically plausible and tested that usually in rats to see how that changes, and then as well to look at those changes, that injury in the organ specifically using histology. We then begin to assess how this biomarker performs clinically, and so we begin looking at measuring this biomarker in patients who are involved in different drug trials. And then another important component when you are using any type of test is that we need to know that that test, or assay in this case, is going to perform consistently and in a similar manner at each place that uses the test, and by each individual or operator who is conducting the test. And then additionally along that is, do we have adequate and relevant data sets? Do we need to move forward with studies that have not already been conducted? And what does it look like? What are the biomarker values for these new biomarkers? What does it look like in healthy populations? And then as well, what does it happen when I say medically significant? What happens to when there is a change that leads to injury? Where is that point of significance in change of that biomarker? So say if normally a measurement is in a range of say 2 to 10 units, when does that change above 10 become injurious to an individual? Is it when you go from 10 to 20? Is it when you go from 10 to 12? And along the lines of ensuring that the test itself works well, there are a number of different components that we have to look at. And that's one, I think, the reliability. Is it consistent across places, as well as the reproducibility within each location? Two words I've brought up a couple times: sensitivity and specificity. And so for that component, does it support then making drug development decisions for the safety of a drug moving it forward? And so as an example, a project that we've been working on for quite some time is looking at liver injury, drug-induced liver injury. And there are the standard biomarker to measure it is alanine aminotransferase. And so first I'm going to talk a little bit before I go into ALT, alanine aminotransferase, about this new biomarker that we are advancing. It's named glutamate dehydrogenase, or GLDH. I'll call it GLDH for short because I don't want to say that dehydrogenase too much. It is an enzyme, and so an enzyme is something that permits a reaction to move forward or catalyzes something. And in this case, it converts glutamate to ketoglutarate, and it does that in the cell component, the mitochondria. And one of the pieces that we look at in terms of the biological plausibility, where is it located? In this case, GLDH is almost exclusively found in the liver. It has small trace amounts in the kidney and the pancreas and the intestines. And another important component for this research is that it is almost there's only a trace amounts. It's not observed in muscle, and that'll become important in the next few slides. And it's also a biomarker that's been shown in veterinary science, and for veterinarians they've used it for a number of years for detecting liver injury, quite a bit in horses and as well in dogs. But there are a few gaps in terms of advancing this for use in humans. And one is that there was not at the time a lot of data that had been published looking at its use in humans to detect liver injury. And so what we went about doing is a systematic evaluation of how this biomarker changed in the liver when people had liver injury. And for this, we used both drug-induced liver injury as well as people who had a liver disease. And so we were working for acceptance of this by regulatory agencies, and then as well educating physicians treating patients about how to use GLDH in place of ALT in the future. And so the first piece that we did is that we took a very large data set. Each one of these circles is an individual person. And what we did is we measured GLDH is here on the x-axis, and then ALT, which is the accepted biomarker to determine liver injury, is on the x-axis, x-axis for ALT, y-axis for GLDH. And what we wanted to see is if in patients that we know have liver injury, does GLDH perform similarly to ALT? And the way that we test this is we look for linearity across the biomarkers. And you'll see that there is a little bit of spread on either side, and that's where we have a little bit of error. But overall, this line we found to be a very strong correlation between ALT and GLDH for injury. And you'll see down here we have these two boxes. And the first box, actually before I get to that box, and then we have this circle down here. These patients all through here were healthy patients. And so this is what we use to establish what is the normal range of for GLDH. And so the cutoff that we found was right around 10. And then for looking at injury, what we did is we know that for ALT, an injury point is at approximately 2 times and 5 times the normal upper value. And then what we did is we went to this linear line and we said, okay, what's the value of GLDH at that point? And we found that to be this number 25. And then as well, a second point, so this is where you start to see injury, and it's a good indicator of injury. And then if you're moving into more severe injury, you have this higher number that's about 5 times the upper limit. And we correlated that to a similar value for GLDH. And so that's great. Now we see this correlation in patients we know. Up here, these patients all had some type of liver injury, people who didn't have liver injury. And we can correlate this straight line and say, okay, GLDH performs similarly in known patients. What about in patients that complicate things a little bit? So in patients with muscular dystrophies, in this case, it's Duchenne's muscular dystrophy. Those, it's a disease that's primarily in boys. And one of the pieces that's very difficult to diagnose with these boys is that their values for ALT, the standard marker, and for AST, another marker of liver function, are considerably more elevated when you compare it to healthy adults and healthy boys. You can see this is, you're talking more than 10 times more than the other value, the normal value. And then this third biomarker down here is creatine kinase, or CK, and that's a measure of muscle function. And so, as you'd expect in boys who have a degenerative muscle disease, something that's causing their muscles to break down, that number is quite high. But then we have these two numbers that are supposed to measure liver function that are quite high as well. And so it becomes quite difficult to, one, diagnose these boys with Duchenne's, because frequently they go to their pediatrician, and their pediatrician looks at these values and says, it looks like they may have some type of liver disease. And then that goes down a whole other path. And so what we've found is that in these boys, if we look at the normal value in adults and the normal value in boys for GLDH, it's around 3. And then in boys with Duchenne's muscular dystrophy, it's around 5. And as I described previously on the last slide, the normal range is between about 2 to 10. And so these boys have a normal GLDH value and then a high creatine kinase, or CK, value. And so right there, we can begin to eliminate liver injury if we were to use this biomarker. And as well, if we look at people, adults, a few adults here, just a very few, a small number, who have Tylenol toxicity. So they took too much Tylenol, which leads to liver injury. We see, so this would be a known injury to the liver. We see an increase in GLDH, which is what we would like to see. We see an increase in the current standards here, ALT and AST, and not a major change in creatine kinase. We did some more studies where we looked at to see if there was a relationship between GLDH and AL and creatine kinase. And you'll see here on the last slide where we had from 0, we had a line that went pretty straight. You'll see here, these numbers don't correlate across, right? If we have a value here of 4, it spreads from normal for creatine kinase all the way up to significantly injured. Whereas with ALT, if we look here where there's ALT on the x-axis and creatine kinase on the y for looking at a correlation plot, you see that this, similar to the ALT and GLDH, there is a very similar, there is a correlation between these two. And so with all of this information in hand, we then began to apply it to an individual case. And this is a really interesting person, an interesting case study. A person presented themselves at the hospital, and they were admitted with extreme muscle injury, which is also known as rhabdomyolysis. And this happens when one does too much exercise or takes something that injures muscle. And when this happens, you see a significant breakdown of muscle tissue, and that leads to a release into the blood of proteins from muscle. And so this person then had biomarkers to assess muscle injury. As I mentioned before, that'd be creatine kinase was measured when they came into the hospital. And you'll see here that this is the creatine kinase, and at the time that it began to increase significantly, so do 2 of the biomarkers that are used to measure liver injury. Here, this is the blue line here, that's ALT, and the red one here is AST. And so, and these are significant changes from what would be normal for a person. And so then you have to decide, does this person have muscle injury? Do they have liver injury? Well, in this case, we also were able to measure GLDH. And at the initial point when the person was admitted to the hospital, the GLDH remained at a normal level. But then, and so that was normal. You see changes in the protein from the muscle. And then they had a hypoxic crisis. So they went into sepsis. And that's when all of your organs begin to shut down, and you have injury across all of your body. And at that point, we do see an increase in GLDH, since there is injury to most of the organs in this person's in this person. And so again, this is kind of furthering the evidence that this biomarker, GLDH, is very specific to when you have liver injury, especially when you combine that with the last slide. And so that kind of walked us through what I do regularly is I work with the scientists to put all those pieces together. And you know, where do we see this going next? Well, one of the big places that you'll probably see and recognize some of these is digital biomarkers. And that's ways that you can continuously collect data on people that is not invasive. And two areas that we see definite potential and my institute are working on right now is in type 1 diabetes, and being able to continuously monitor their sugar levels, because people with type 1 diabetes are not able to regulate their sugar levels very well, and so they receive insulin as those levels change. And so being able to monitor that without continuously taking a blood draw or a finger prick is very desirable, because that improves their how they live. And then as well, in people with Parkinson's disease, is looking at measuring different pieces of how they work it during the day. So it has to do with how they walk, how their arms move, if they have tremors. And so using things like an Apple Watch or a Fitbit, you can, throughout a day, continuously monitor, in the case of Parkinson's disease, someone's gait or how they walk, their balance, what their spatial memory looks like. And then, and so, you know, looking at spatial memory, and then also looking at how they can tap and what type of dexterity they have. And finally, looking at what their speech is like. And so that would then be continuously monitored throughout a day, and would reduce potentially the number of visits to the hospital. But this information would be shared with their physician to monitor how they are, how the disease is progressing or is not progressing due to a new treatment. So with that, I thank everyone for listening to me right now, and I welcome any questions.
That was great, Nick. Thank you so much. Nick, can you hear me?
Yes, I can.
Hi. Thank you so much. That was great.
Oh, yeah. No, I'm happy to be here today.
It's fascinating. I think a lot of us take medications here and there and don't think a lot about what goes on before they end up in a pill bottle. So it's fascinating to hear about your research.
Yes, definitely. It's one of the, you know, where I work is not something that most people, you know, I didn't necessarily think about before I moved into what I'm doing today.
That's awesome. Our first question really kind of relates to that. This is a general question we ask all scientists. How might someone like yourself, who is a young scientist right now, get into the field that you are in?
Oh, that's a great question. Thanks. The first piece, I started out here as a project manager, and then I also had a strong background in being able to talk with scientists. And so around the project management, I think as you're going through your education, look at ways that you can be as the most organized as possible, and also how well you communicate with people across different media. For my job, I work usually in an office with people within my company, but then as well, we have a large number of external stakeholders. And so being able to talk to them on both the phone and in person, which are different skills, are pieces to definitely work on. And I continue on that piece every day. Every day is a little bit of practice, right? But really, more than anything, it's looking at how organized you can be, and putting together, looking at sequences of what you do, and being able to track that. That's how I moved myself into project management here.
Interesting. It sounds like quite the journey. So we have our first question here from an audience member. Are there other diseases other than muscular dystrophy that GLDH can be used to help evaluate or diagnose?
Yes. So well, what I should take a step back. It's a great question. So with GLDH, it is something that is used to eliminate one potential type of disease. So in this case, you have with muscular dystrophy or other muscle injuries, you have several different types of information coming in that are difficult to interpret. And one of those is measuring your liver enzymes, the ALT and the AST. GLDH, since it doesn't change in those patients, it eliminates that the liver has been injured and permits you to look at other issues. It can act in that same way for most types of muscle disease. Another area it can be used is to monitor in people, say, with different types of hepatic diseases that may have other complicating factors that lead to difficulties in monitoring the normal markers of liver injury.
That makes sense. Thank you. So I had a question myself. I wondered, you talked a little bit about the partnership between the US FDA, the European Medicines Agency, and then the PMDA from Japan. Do these companies specifically, or do these countries specifically have similar medical regulations, or are there reasons specifically that they work together?
So each agency operates independently. And they all have similar, I think, objectives, but slightly different approaches. In the US, we keep a lot of scientists are employed by the FDA to not only review new drugs, but also to conduct basic research. We also use them for monitoring food, right, as well as cosmetics. And so they have a much broader set of pieces to evaluate and monitor in the U.S. In Europe, the EMA is solely focused on approval of drugs and then approval of devices. And in Japan, it's actually a little bit more similar to the U.S., although it's integrated with a few other agencies.
Interesting. And so I wondered, when thinking about the sort of more severe impacts of some of the drugs or the components of the drugs that you're studying, what are some of the ethical concerns behind that, and how do you decide when an impact might be too severe?
So that right now, it's I think there's two big pieces I think to consider when we're looking at severity of injury. One is what you're measuring actually injury, right? Is it a true injury? And some of the ways we monitor for those right now don't tell us if there's true injury. I think in the case with the muscular dystrophy, you see those elevations in liver markers, and yet there's no liver injury. And so when we make those decisions, we base the changes in these markers to really drive when something is too high of a risk. For our work, I think the other ethical consideration I think that is always on the tip of everyone's tongue is probably part of what you're bringing in here is the use of animals in these studies. It's something that I think everyone would prefer to not do. Currently, it is the primary method for initially assessing injury or efficacy of a new drug as we move it forward. though there is a lot of research right now that is looking at alternatives to that, such as taking cells and having in vitro cell systems where you incubate cells and then test them with the drug and look for injury to those specific cells, or even building up what are called microphysiological systems, which are little mini organ systems. And so while it's not a person or a rat or a dog, it's taking cells from a human's a liver, a heart, and say other organs, and putting that into a system that interacts similar to a full organism without actually testing it in a human or a rat.
That's fascinating.
Yeah, and I have one last piece there too. Sorry. That I think this is another really cool thing is that when we have these biomarkers that are more sensitive or more specific to the type of injury, we have seen once they're accepted for use in rat studies that you don't have to do as many studies. So you actually reduce the number of studies that are necessary because you get more accurate results.
Mm-hmm. Fascinating. Sounds like the future of medicine. That's really fascinating.
It's definitely, yeah, and it's moving towards some of the personalized care, right?
Yeah, that's great. So I think Lara has a pretty close follow-up question to the one you just covered, but specifically, how do you determine when the benefits outweigh the risks of possible injuries that a drug could cause? Or is there a certain measure there?
Thank you, Lara. It's a difficult one because it depends. There's certain ways we're going to weight that type of a decision. And so it depends on the severity of the disease and then as well how effective the drug is, and then looking at what types of risks or injuries you expect from that. And so I'm going to take a couple extremes here. So say you have someone with a very advanced stage of cancer. We're willing to take a few more risks to remove that cancer. In the case, I think everyone knows that, with chemotherapy, you usually lose your hair. And so that's a risk that we're willing to take in order to save the whole person. Whereas, say you have just a minor allergy, just kind of hay fever every year. We wouldn't be willing to lose our hair so that we didn't sneeze.
Mm-hmm. That makes sense. So it sounds like on a case-by-case basis, you're kind of weighing the specific situation.
Definitely, yes. It doesn't have, a set line in the sand that you can make that decision. It's every time, and it's looking at everything holistically.
Great. Well, thank you, Nicholas. And thank you for all of the work you're doing to keep all of us safe and healthy. Thank you so much for giving your time to our audience to tell us about what you do.
Yeah, thank you for having me here today.
All right, so if you have any other questions for Nicholas King, you can always leave them in the comments. We'll put up the archive of this video afterwards. And otherwise, thank you so much, Nick. I hope you have a good day.
All right. Yeah, you too. Bye-bye.
Bye.